breakthroughs in fish health and nutrition, and probiotics, bioenzymes, and algal culture, algal-based feeds, all these sectors, and which not only diversifying the field but also reducing the aquaculture footprint and reducing the antibiotics stress and many things. While genetic tools like CRISPR-Cas9 and other many marker-assisted selections and all these tools are improves the breeds or the stocks which are disease resistant and climate resilient species. These advances not only improve the productivity.
Sir, sorry for interruption. Sir, slide mode कर लीजिए sir PPT.
Yes. Okay.
Presentation mode.
Okay.
Okay, sir.
So I am saying that not only it is increasing the disease resistance and climate resilient species. These advances not only improve the productivity but also minimize the environmental impact. As these innovations move from laboratories to farm, they create a ripple effects in the markets. The eco-certified seafood, bio-based feed, and alternative feeds, and the circular production and circular economy-based production systems, gaining consumer trust and opening new economic opportunities to the stakeholders or for the stakeholders. Then the waste is being repurposed into valuable resources, and efficiency is driving the profitability. With this background, myself, Dr. Aditya Pratap Acharya, Assistant Professor from West Bengal University of Animal and Fisheries Sciences. Today, I will discuss the topic from molecules to markets. So, in this topic, I will not go in detail, but I will give a crisp idea or crisp view about the present scenario and what are the present innovations in the aquaculture, so that a complete overview can be gained. So, the background is aquaculture is the fastest growing food production sector globally, and however, sustainability, as the speed is high, so there is always a chance of accident. So the sustainability acts as the accelerator and brake or clutch. So in this way, the the fastest growing system should be sustained, so that there should not be any sudden crash. So I will, for the better understanding, I just made one ABCD framework or the alphabets of challenges. By addressing these alphabets or by learning about these alphabets of challenges, we can understand the sector well. And we can see what are the management options and what are the challenges. So why it matters? Because it is not only a commercial point of view, it is very much important for the point of food security. Most of 51% of the animal supply are from aquatic sources, and the environmental, which is due to the near future, the population may reach up to 9.7 billion in the year 2050. So the production need to be increased 25 to 70%. So these huge production will ultimately create pressure on the environment. So we have to take proper steps or precautions in appropriate way, so that it should be sustainable. And at the same time, it should be feasible and economical also. So these are the three things today I will discuss. These are the molecules and the foundations. Understanding these 3 points, we can discuss how the biotechnology are reshaping the Indian aquaculture or global aquaculture industry. So genomics, in the genomics, we are discussing about the DNA sequencing and marker-assisted selections, which improve the better fish trend, faster growth rate, disease resistance, and field efficiency. Nutrient optimization that helps identify and reduce the anti-nutritional factors, because when you diversify the feed, many of the feeds contains the anti-nutritional factors. So as India is a carb country, it may not require the fish meal. But meantime, we can depend upon the soybean, but the soybean is a plant source, so which carry the many anti-nutritional factors. So how and which way you can reduce the anti-nutritional factors and health management. as the intense culture practices is followed, so which create the health issues. So these are the three pillars: genomics and breeding, nutrient optimization, and health management. So these are the challenges why the system. I mean, these are the challenges for which we need to address these challenges very quickly for the sustainability, governance and planning, weak policies. Sometimes the biosecurity, someone is following, someone is not following, someone is following in when there it is nearby cities and urban areas, but the same is not followed by the for a rural farmers. So this should be uniformity, and the coordination should be strong. Resource and ecosystems. Pollution. When pollution comes in urban or rural areas, it will affect in long term for all the population nearby to these areas. So feed inputs, sustainable sources are needed. Biosecurity, welfare, disease, and AMR, antimicrobial resistance. It is a very burning issue, and it is in the One Health concept. This issue is the major concern. Then climate change and environmental factors, social and economic disbalances, and markets and value chains from which it is created, the source, origin, and the way it is moving ahead, like from the village to export. So during the value chain, what are the constraints, what are the risks and associated with it, we will discuss. Then capacity and innovation, how the training, research, and awareness can improve our export potential, so that although 30 million people are dependent on aquaculture sector, so more people will involve in this and more profit we can take from this sector. So first we will move ahead, we will understand what is a aquaculture. So whenever like agriculture, whenever we are talking about aquaculture is nothing, everything is natural, but the thing is our intervention for better enhanced production, or by regular stocking, feeding, and protection from the predators. If we are doing these three things, in natural condition, what we are doing, we are just simply go to the river or the sea. We have the nets and other accessories. We'll collect the fish directly. But there we are not thinking about the restocking or any feeding strategy. But in aquaculture, we are providing feed. Then we are protecting them from the predators, and we are collecting or taking the profit. So this is the aquaculture deficit. So what is sustainability? Sustainability is defined as the responsible practice. The term responsible practice. It is not today I am taking profit means I will stock many more stocks, and I will pollute the ecosystem, pollute the water body, pollute the through enormous use of antibiotics, enormous use of probiotics, and many things. So everything should be in the limit. So responsible practice of farming aquatic organisms such as fish, shellfish, and aquatic plants to meet the current needs in such a way that the current needs should be fulfilled, but either way it should not affect the future generations' need. So this is the sustainability. The key principle includes minimizing the ecological destruction, ensuring animal welfare, and promoting long-term economic stability. So I have already discussed about this thing, that in the 2050 the population may reach 9.7 billion. That time we may increase our food production. We have to increase our food production capacity or aquaculture production capacity to 25 to 70%. So it is a huge burden. And the best thing is aquaculture is so popular, and so the pace is so good that in 2022 the aquaculture production surpasses the capture fisheries. So now we are more production from the controlled environment, and which is simply doubled. If you see the production, it is from 1990 to 2020. It's simply doubled, which is from 81.6 million metric ton to 164 million metric ton now. So the ABCD challenge, which I have told, for simplify animal, here the animal is the fish species. So whether the species we are, or the stock we are maintaining, that is a good stress resilient, eco-friendly, I mean can sustain the climate change, and its survival should be good, its nutritional adaptability should be good, and it should be ethical. We should not create such a different thing which is not ethically or maybe not accepted by the consumers. Then biosecurity and biodiversity. So control of invasive species. Like one example I will share here, Thai magur. You may know the African magur, whose growth rate is tremendous. It can go within 6 months. It can go up to 1 to 2 kg or 3 kgs. Whereas in our natural magurs, Clarius batratus, so that's growth rate is normal. So farmer in the grassroots level, they will not understand, oh, this fish is growing in the faster way, we'll culture this. But the thing is, that is not the native species to our country. So that is a foreign organism. So when it invasive to our ecosystem, it will destroy the ecosystem balance. The gene pool will be diluted or polluted. It can bring some other diseases which is not previously present in our ecosystem. So these things, biosecurity and biodiversity laws, should be monitored properly and carefully for sustainability. Then cost and climate pressure, like I have discussed about the soybean meal and many other things, rising feed cost, or alone feed cost is compromising around 60 to 70%. So then infrastructure, investment, climate risk, all these things need to be taken care for sustainability. Then disease and disaster risk. Disaster risk for a country like India, always there is a disaster. So how to checkmate that disaster, how we can construct our ponds, how in which way. So that is the part. Then environmental footprint, whether it creates pollution, it prevents habitat destruction, carbon emission, these things need to be taken care. So here animal fish welfare, genetic quality, and broodstock management. So what is the genetic purity, or what is the intention of creating one particular commercial breed or strain? So and how it should be, how the broodstock should be managed so that minimum inbreeding will be there. So that should be taken care. Stocking density, how much fish should be stocked in that particular aquaculture system. Whether we should not that should not be over dense stock, or should not be less stock. If you are overly stocking, then the chances of disease spread will be high, and if you will stock less, then the chance of we are not properly utilizing the resources. Stock health and physiological stress. Stock health means from where you are bringing the stock, whether it is a SPF, specific pathogen free stock, or it is a specific resistant strain, or it is any specific particular characteristics, and whether from how much distance you are carrying. You are carrying from the nearby farmers, or you are carrying from far away. So all these things need to be taken care. Then biosecurity, I have already explained. Pathogen exclusion and quarantine protocols must be followed for sustainable aquaculture. Then seed certification and traceability. So what this is seed certification means, I am an aquaculture farmer or a hatchery producer. I am saying that my seeds or my larval piece are some pathogens free, or whether I have I am creating one particular strain which has better growth rate, like joint pirohu. They have prepared for selective breeding. So they can issue a certificate that these strain is this is the joint pirohu strain. So it has a better performance than the normal room. And traceability, whether once you expose your product to the market, we should train, or someone should trace your product. If you are not able to trace your product in the same way, like duplicate packets are available with the same branding, same certification. So what if you can trace your product, the trust will be increased. Now, no, there is no dilution with my product, and it is properly distributed in the market. Then farm sanitation protocols should be followed, movement should be controlled, surveillance and yearly warning systems. Surveillance, each time it should be disease monitoring, and everything should be properly weekly monitored, so that there should not be a sudden chaos that there is a breakout of some disease. So that should not be. If your surveillance and yearly warning system is there, so through that system you can know whether there is any risk or not. Rising feed and energy costs, so we should focus on alternate feeds. and alternate feed resources. Disease outbreak, we have already discussed about this. The viral, bacterial, and parasitic infections that should be biosecure, and the quarantine protocol should be followed so that the existing disease will not spread to the other farms nearby. So these emerging pathogens, intensive system, antimicrobial resistance risk. Some in aquaculture, what happens? Many farmers are not aware, and some company person is going and they are saying you just use this antibiotic, but there is not recommended. There is one recommended level for 1 hectare area. When you are discussing about the aquaculture, it should be cubic meter. So without knowing the standard appropriate concentration, if you are throwing dumping the antibiotics, so it will create health hazard. So in this way, and environmental impacts I have already discussed. So to mitigate these challenges, biotechnology is the bridge. So how this bring to the ponds? I have told you that genetic breeding or the marker assisted selection, where we particularly selecting certain genes and creating one strain and release that strain into the pond. That pond, that the efficiency of that brood fish is so high so that it can be it can reach up to the market, and which is economically viable and accepted by the consumers. So this is how the genes to the pond and laboratory to the market. So here the functional genomics play the role, and the three major pillars are now in the current trends are marker assisted selection, CRISPRs, and omics approach. So innovation in fish breeding. If you will see what are the innovations in previous, there are in genetics, in terms of genetics, there are selective breeding. But now in the era of biotechnology, we are not much dependent on the selective breeding. Now we are doing the selection through marker assisted selection, not by the traditional phenotypic characters. So, and genetic manipulation like gene editing. For gene editing, there are Tolen and ZFN and CRISPR-Cas9. But Tolen and ZFN are outdated, and gene editing tools like CRISPR-Cas9 is the most accurate and most reliable method till now. Then monosex production. These are the methods. So what is marker assist? What is a marker? Marker is simply a markers can be a molecular marker, or so the molecular markers are DNA marker, and it is a neutral marker, it is a codominant marker. So like the most common, that is another part. But what are the markers? But the marker is a short sequence of the DNA fragment through which we can ascertain certain location of the genome that this marker is present, like a landmark. When you are moving in a road and you are getting that this is 20 kilometers away, Mumbai is 20 kilometers away. So that pillar is the marker. That 20 kilometers away you will get Mumbai city. So here in genomic, there are certain markers, genomic markers. Through that markers, we can ascertain that certain location of the gene, whether the gene is located, how much distance from the marker. So in genetics or in biotechnology, these two markers are tremendously used. One is SSR and one is SSR is simple sequence repeats or microsatellite markers. Another one is SNP, single nucleotide polymorphism markers. These are the markers which are tremendously used. The marker assisted selection transform aquaculture breeding from phenotype based guessing, that we are guessing, oh, this fish is growing faster. So we have to observe, we have to wait for one generation. Till the fish should reach, we can 6 months to 7 months, we have to wait whether the fish is growing or not. We don't know whether the fish grows due to environmental factors or due to the genetic factor. If the fish is growing due to the environmental factor and you are choosing that on the basis of genetic factor, so again you will be failed. So from guessing to the genome guided precision improvement, this is the advantages of marker assisted selection. So why these marker assisted selections are important? Because there are certain, these are the traits which are most demand and most needed, but these the heritability of these traits are very low. What are the traits? These are those traits that are influenced by environment strongly, like disease resistance, survival rate, stress tolerance, reproductive success, and behavior traits. These are the traits which are very low heritability and difficult to measure to normal or guessing through the normal phenotypic way. Hard to measure traits like feed conversion efficiency. need individual feeding time. We have to keep the fishes and you have to feed, and after 30 days, 60 days, 90 days experiment, you have to prove whether that fish is growing with that feed or not, which is again time consuming. So instead of these methods, we can do the marker assisted selection. We have to identify certain markers and we have to identify certain genes. If that gene is present, then you select the fish. If that gene is absent, then don't select the fish. As much simple as. So these, the hard to measure traits are fillet quality, feed conversion, disease resistance, metabolic efficiency, and immune resistance. These are the traits. So how to develop a marker? Like there are many markers, but I will simply explain the how to develop a microsatellite marker. So the left side figure, you see that is the genomic DNA, a long loop, and you have to the blue, red, and markers are the fragments where the microsatellites are present. Microsatellites are the simple repeats like A, T, 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 G, 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 C, C. Some are 6 to 20 repeats, or sometimes 6 to 100 repeats. So these 2 to 100 repeats. So these repeats, if they are present, these are can be act as the markers. So simply you isolate the DNA, collect the sample. I will. This is the again. I will back to the picture. First, you have to collect the fish specimen, from which fish you want to develop the marker, like a katla, rohu, mrigal, or if in agriculture you know which tree or which plant need to be for your selection. In that case, you collect the sample, isolate the DNA, check the quality, then fragment it. Site selection, whether the quality is good, then fragment it. After fragmentation is depend upon the on which platform of sequencing platform you are going to use, whether it is a nanopore, whether it is a ion torrent, whether it is a Illumina, what kind of platform you are using. Accordingly, the sites should be fragmented. This fragmentation can be done through mechanical method or enzymatic method. Mechanical method by sonication. Just there is a machine sonication, so the sound effect you can break the DNA sonic waves. Or in the enzymatic method, simple, there are so many enzymes available: blunt end cutter, frequent cutter, and rare cutter. So depending upon the need, you use bring that enzyme and incubate for 5 minutes. That your DNA sample will be degraded into the small fragments. Then labeling. If you want to direct, you can go directly for sequencing, or you can label it. Labeling is you're actually getting chance of getting the microsatellite marker will be high. So label with the probes. Probe is the single stranded DNA, which single stranded DNA which is labeled with certain label. Okay. Then site selection, then amplification. Just after you site, just amplify the product and the library quantification and probe copy. I will describe. If it is difficult, should I explain it, or I should not explain it? Anyone? Hello? Hello? Anyone is there?
Hello.
Should I explain it details? I mean, if everyone's background is okay from biotechnology related, then I will explain. Otherwise, I will skip. So anyone, from the participants, anyone interested? So I should explain all these steps or not? Okay, let it be. Okay, I will simply explain. Shortly, I will explain. So in this method, you have to. So this is.
Sir, go for presentation mode, sir. Presentation mode, sir. Presentation mode, please.
Yes, yes. So simply you choose and you select.
सर एक क्विक आउट हो गया वो प्रेजेंटेशन मोड नहीं है सर वो स्लाइड आपकी विजुअल नहीं है सर
अभी हो रहा है
नहीं
Now?
सर फिर से शेयर करना पड़ेगा आपको आप स्क्रीन से बाहर हो गए हैं
Okay. Hmm. Okay. Okay, now it's visible hopefully.
हाँ विजुअल सर, प्रेजेंटेशन मोड सर कर लीजिए।
Okay.
ओके सर।
So seed certification scheme for sustainable aquaculture, and in India it is under the name Safari. Safari, it stands for superior quality seed product for human consumption. At global, if anyone want to get this certificate, any aquaculture farmer or producer, they can go to the website, Entrada website, and get. There are some global certification schemes also, that are eco-labeling, branding, and certification. Eco-labeling is nothing but a eco-label. It's a tag that placed on the product that certifies that the product was produced in the environmental friendly way. That means your product, if you are making in a organic way or in a eco friendly way, then you will apply for this tag, and due to this tag, there is the more visibility in the market, and consumers will prefer this product. Branding, like some salmon, in the salmon industry, it is most common that if these companies, Aqua Advantage Salmon and their other salmon factories, so if you are getting this branding certificate, your product has a brand name. Like if you are Allen Solly product, you will prefer Peter England, you will prefer. So this is the same way your fishery product will be branded. Certification, if you will get any certificate from any NGO or aquaculture society, or there are many societies, you can ask for certificate and your product value will be increased. So these are the certain schemes through which you can apply. So like seed certification. So in this seed certification, you will simply, breeder will produce the seed, field in the team will visit your farm, and seed they will sample, and the DNA, through the DNA samples, DNA markers, they will test your sample. If it is okay, they will give you a tag and a certificate. Then seed traceability, which I said, from the farm level, they will collect the sample and test it, and they will update in the website. And so anyone, anywhere with this tag can identify your product. So the best aquaculture practice, BAP, is the world's most trusted and comprehensive proven third-party aquaculture certification program. So which is started from 2002, and this certification is valid for these categories: shrimp farm, tilapia, channel catfish, pangasius, seafood, and feed-based and salmon farms. Which test to be done for antibiotics? If you are going, if you are challenging, if you are saying that my product is a, now if you want to sell your product abroad or export your product, there should be antibiotics free. These antibiotics should not be present in your sample. So these are chloramphenicol, these are the least, tetracycline, and others. So this test can be done through ELISA, microbiological inhibition test, LCMS, or HPLC. So if you are qualify these tests, then your product will get the certificate. Test will be like, if you are challenging that my product is SPF, specific pathogen free, then these are the OI listed diseases that will be checked, and you will get the certificate. White spot syndrome, Torah syndrome, and these are the list of syndrome. Then we will discuss about the gene editing tools. So already I have discussed, these are the CRISPR tolerant and RNA interference. I will discuss about the CRISPR. So CRISPR technology is nothing but it is consist of two molecules. It is one is Cas9 protein, which is nothing but an nuclease, like in any nuclease enzyme which cuts the DNA, and guide RNA. Where to cut? The guide RNA will direct the along with the Cas9 protein. The Cas9 protein will be guided by the guide RNA. Where to cut? So this is the concept. You have a nuclease enzyme, and you are like the letter, you have writing any letter, and on the above letter there is the address. Address is the guide RNA, and inside whatever you are writing, that is the Cas9 protein. So combining both these two, you can you can send your letter to anywhere. Like through this method, you can edit the gene. Okay. So this is the genomic DNA, and a palm sequence nearby the PAM sequence, the Cas9 will cut the DNA fragment and destroy the your target. It can be knocked down or knockout. Knockdown means it will simply abrupt the gene so that the protein will not produce, or knockout means it will totally disrupt. Some lychee production will be there, or otherwise there will be no production. So through this method, these are the traits improved by the genome editing in aquaculture. So how these are the target region to alter the pigmentation pathway, increase the growth rate, enhance immune response, feed conversion efficiency, nutritional quality, and sex control. And where the successfully experiment was done for the species like goldfish, common carp, tilapia, Atlantic salmon, medca, and all these. So I have already give the references of Zhu et al. You can refer this paper. Like if you go through, there is a simple comparison. If you want to, in which way you want to produce, if in the traditional way, it is very time consuming, 5 to 6 cycles of selection needed, and multiple backdraws you have to do, time consuming, many undesired genes along with during selection, many undesired genes also can grow, which is time consuming process. Whereas you in this right side B picture, you can see there is a precision breeding, CRISPR-Cas9. You select the gene and use this CRISPR-Cas9 technology that will disrupt the gene, and you will get your product in one generation. Then we'll discuss about the omics approach. What are the omics? There are 3 omics: proteomics, transcriptomics, metabolomics. What is omics approach? Studying about the whole system at the molecular level. So what is proteomics? Proteomics is the large-scale study of the complete set of proteins, proteome expressed by the cell at a particular time. So the technology involved for this 2D gel electrophoresis, LCMS, MIDTOP, Western blotting, protein microarray, and etc. So application of these proteomics are personalized medicine, biomarker. In case of aquaculture, the host pathogen interaction, climate adaptability, environmental stress management, and food safety and allergen detection. Because when you are develop a strain to gene editing, we don't know whether the product will contain some allergen or not. So in that case, these 4 states are important in proteomics point of view: host pathogen interaction, climate adaptability, environmental stress, and food safety and allergen detection. Okay. Then transcriptomics, the comprehensive study of the entire set of RNA transcript. So the key techniques are RNA sequence. The common technology used here: RNA sequencing method, microarray analysis, real-time PCR, single cell RNA sequencing, or not the These are the technologies for which one can do the transcriptomics study. Application of this transcriptomics: gene expression or profiling tool, genomic tool, disease diagnostic tool, drug discovery, and toxicology study. The transcriptome can be utilized for this kind of study. Then metabolomics, the systematic analysis of all the small molecules, metabolites present in the biological system are called as the metabolomics. Here also LCMS, NMR, CEMS, and GCMS used to do the metabolomics. Application of metabolomics for hatchery production. That means metabolomics are like proteomics and metabolomics. Proteomics and transcriptomics. Metabolomics is a little bit, the data generation is very low, and it is very friendly, and one can the technical demand is less also in metabolomics study. So how this can be applied for the hatchery production? Hatchery production means the seed production. Aquaculture larval rearing is the most challenging part for many species. Production yield, sometimes you will get 90% of mortality. Your whole batch will be died. So in that, and there is an unknown reason for that. So there is no previous record also, and it is very difficult why one batch is suddenly died. So it will give a great loss to the farmer. So that time metabolomics can improve the mechanistic and functional biochemical information from which we can analyze the remedial strategies for poor hatchery performance. You can know the cause why this batch is failing. Like sperm fertility, if one particular metabolite is present, then if that bioindicator or biomarker is used, If that is present, the sperm is fertile or less fertile or high fertile, you can assess. Assessing the oocyte quality and predicting the embryo viability, whether the embryo is viable or not. So this can be studied. Identify the coordination of metabolic stress during selective breeding for stress and resistance. Then higher survivability. This can be application of aqua nutrition and aqua diet. So these metabolomics can assess the positive and negative effect of food deprivation. When there is no food, how the fish will react. Interaction between diet and environment. One different diet you are giving at a 30 degree, 40 degree, 50 degree environment. So how the fish is reacting. Effect of nutrient supplement in one different kind of nutrient you are supplying. What are the effects you can study by assessing the metabolomics. Effects of dietary protein substitution and utilization in salmon and carp. Effect of contaminated and decontaminated fish meal in feeding. Effect of using reduced fish meal. Effect of replacing fish oil with vegetable oil. Effect of newly introduced plant-derived contaminants. So all this can be studied through this metabolomics study. Then feed innovations. I have already explained that 60 to 70% of the total cost, input cost, is only through the feed. So industry is depends on conventional feeds like fish meal and soybean meal. So which are again the soybean meal has the human consumption. Thereby, if the market price is increasing day by day, and which is not a dependable source, protein source for aquaculture industry. So therefore, one high quality, affordable, and sustainable alternative feeds should be ingredients must be find out. So I have already told that carp country like India, commercially cultured carp species do not require marine derived proteins. So what are the alternatives? So the other alternatives like MOC. One example I am taking about, there are many alternatives, but out of which I just want to take one example, mustard oil cake, which is very good protein source, 20 to 37% of the protein. But the thing is, the anti-nutritional factor is which is tannin and saponin, which is contributed around to 89%. So if we have a method to reduce the anti-nutritional factors, because mustard oil cake there is no demand for mustard oil cake for human consumption. So the rate will not increase in the future. So that's why it may be an alternative for sustainable aquaculture as a alternative feed source. But how to reduce the anti-nutritional factors? These anti-nutritional factors can be reduced through fermented process. Fermented is the by which way nutritional value and digestibility safety can be increased. So how this works? Introduction of beneficial microorganisms, including yeast, filamentous fungi, and bacteria into the substrate to initiate the anaerobic production. If you are you are producing something anaerobically, that is called as the fermentation process. So it will simply break the complex organic compound into a simpler ones, for which that will be more palatable, more the shelf life will be increased, the toxic compound will be degraded, and which is helpful for the aquaculture animals to eat. It is one side. Till now we are discussing about the aquaculture and growth related performances, and now we will discuss the ornamental industry, where in case of ornamental fishes, the growth is not a paramount. Growth is not a critical factor. The critical factor is the coloration. So how we know the coloration can be improved by carotenoids. The carotenoids can be divided into two groups, like synthetic carotenoids and natural carotenoids. The synthetic carotenoids are lab made, and if you are using these carotenoids, that creates many issues and that are not dissolved, and that will be that will stay in the ecosystem sometimes, and which create many another health hazards. Whereas the natural carotenoids, along with supplement, it is act as sometimes it is growth promoter, sometimes it is give some resistance. So that's why people, and in this era of eco-labeling, if you will sell your product with the eco-label that I have this color is obtained by the natural carotenoids, through the natural carotenoids, people will prefer your product, or people will take your ornamental fish quickly. So that is the benefit, and that is eco-friendly also. So that's why the ornamental fish business offer high profit despite its slow trading. Like India, we are nowhere. We have our rank is 31st in the ornamental fish export, and we are only exporting 0.32% of the global market. But which will again increase. The present ornamental fish market is around 11. Uh, the US dollar, it is around 12 billion market. So there is a tremendous potential of India. We can increase this ornamental fish export, and we can increase more income. So carotenoids are often these natural carotenoids obtained from plant parts, microalgae, seaweeds, and crustaceans. But again, the microalgae and crustaceans, the crustaceans are again sometimes it is very difficult to get, and although it has a good potential, but now people are thinking about the plant derivatives, because variety of plant sources like leaf, fruits, seeds, flowers, these are also used for coloration or carotenoids. As India is producing so much of papaya, and so the papaya peel is a good source of carotenoid which can be used, and which has many other bioactive compounds. So this is one of the studies where we can use the papaya peel as a natural carotenoid source to enhance the coloration of the ornamental fishes. So we discuss about the aquaculture industry, ornamental industry, and about the whole aquaculture production. Now we will discuss uh, in the previous two topics we discussed the fish, about the fish. Now we will talk about the environment. So whether your environment is in which condition you are growing your if your environment is polluted by antibiotics through AMR, AGR, so that should be checked, check and balance. Otherwise, sustainability in the we are producing now, but we are unable to produce in future. So for which the metagenomic study is the best gold standard. So where you simply collect the DNA and sequencing it, and do the phylogenetic analysis and functional classification. Phylogenetic analysis means who are they, who are the relatives, what kind of bacteria, what kind of microbiomes are present in the soil, water, and fish, in fish gut. Okay. Whereas in functional classification, what is the genes, what are kind of genes are present, and how they impact the living being. So here we can analysis one polluted soil. You take one polluted soil sediment and non-polluted soil sediment, and analyze sample metadata processing and metagenomic sequencing, environmental metadata. So if we compare, by comparing these two data, biological diversity analysis and species distribution, what kind of species, what kind of microbiomes are present, we can say aquatic health condition and toxicity level. Like heavy metal is there. How we will identify the heavy metal? If heavy metal related genes are present there, we can say there are certain heavy metals is present, because we are using so much of antibiotics, so much of manure and all those things. That may be the reason. So the aquatic health, the soil health, the everything, and the toxicity level can also be analyzed through the metagenomic data. Non-indigenous species environmental impact. If the eDNA, through eDNA, we can analyze the whether there is presence of any non-invasive species or not. Then if excessive nutrient you are using, what kind of changes in the soil, that also can be diagnosed. So these are the softwares, these are the links and softwares through which the data can be processed. MGA and MGRAST. MGRAST is the most common, IMGM and camera. Kaichu, these are the common use for this. And the software packages, all these things, CLC is the free workbench. You can through CLC, you can use many of them free. So one can visit the website and through the tutorials can analyze. So in conclusion, you can say sustainable aquaculture requires integrated management of animal welfare, biosecurity, cost effectiveness, and disease control and environment protection. So finally the ABCD framework provides a structured approach for research, policy, and farm management. Okay, these are the references. Very few references, because I know if you these two papers, 2-3 websites you will visit,